Hello, everyone. Welcome to the Academic Resource Center's NAS-2 Workshop. So first, let's go over what the NAS-2 is. The NAS-2 is the exam that can be taken as the prerequisite requirement to take anatomy and physiology, also known as Bio 141, in place of Bio 101. So the test covers concepts from Bio 101 in order to prepare you to take Bio 141. And if you pass the test, you are able to enroll in Bio 141 without actually having to go in and enroll in Bio 101 and take the actual course and pay for it and everything. This exam is very helpful to you to be able to test out of Bio 101. Ready? And first, I just want to talk about the Canvas course that you can enroll in. It's free to enroll. It's open to any student. You can enroll in this course, and it has a bunch of readings, has a bunch of quizzes, practice quizzes for you to take in order to prepare for the exam. So if you enroll in this course, you get all of that information, study objectives, study materials, and everything in this presentation was somehow taken from that study course, so that you all can prepare for that easily. Ready? So just an overview of the exam. There are 50 questions that includes multiple choice and true and false. I think it's mostly multiple choice though, not as much true false. It is 90 minutes long. You have 90 minutes to take it. It is closed book. You have no resources available to use. You can't use notes, textbook, anything like that. And the material is split between the different topics in the Canvas course, which are also covered in this presentation. And you have to take it through the Bright Point Testing Center. So either on the Middle or Chester campus, you have to take it through them. You have to have a minimum score of 70% to enroll in Bio 141, but if you do not make that on your first try or even your 10th, you can take it an unlimited number of times within a 24-hour waiting period. So take the exam, see how you do after studying, and if you do not make a 70% or more, you have to wait 24 hours and you can take the exam again. But in that 24 hours, I would recommend studying some more, seeing maybe what you struggled with on the exam and going over those topics again. These are just a general overview of the topics that are covered on the exam and in the study course. So we'll go over atoms, chemical bonding, water, solutions and concentrations. I'm not going to read everything on here. You can pause the video and take a quick look at this, but these are everything that we will cover in this presentation. But if you also want more information, please go and look at the Canvas course, and I definitely recommend getting more information from the Canvas course. Ready? So first we'll talk about atoms. All matter consists of atoms. So everything that we see is made of atoms, even us. Each atom belongs to a specific element. And the most common elements in the universe are oxygen, carbon, hydrogen, nitrogen, calcium, and phosphorus. Trace elements, such as iron, are needed for life. We need iron in order to live. Atoms are made up of 3 subatomic particles: the proton, the neutron, and the electron. We'll talk more about those in depth in a second. And the atom consists of 2 regions: the nucleus, where the protons and neutrons are found, and an orbiting cloud, where electrons are found. So over here in the image to the right, we have a picture of an atom, it's a very basic atom. In the center, everything in the yellow circle in the middle, the blue dots are neutrons, and the red dots are protons. And then they're enclosed in the yellow circle, which it represents the nucleus in this case. Everything around it is the electron cloud. So the green dots represent electrons, and the gray ovals represent the kind of like the paths that the electrons travel on. However, does not look like this. They're very random. They just don't hit each other. They're not, going in a complete oval. They just are showing you this to show that that's what exists outside of the nucleus. Next, we'll talk about subatomic particles. So like we said before, we have protons, neutrons, and electrons. Protons have a positive charge. Neutrons are neutral. They have no charge. Electrons have a negative charge. The number of protons in an atom determines the element. That is known as the atomic number. So over here we have helium. Its atomic number is 2 because it has 2 protons. The atomic mass of an atom is the average mass of all isotopes of the element. So you can also find that by finding the sum of the protons and the neutrons in the atom. So in this case, 4 would be the atomic mass number of helium. And then outside of chemical reactions, the number of protons and electrons in an atom are equal. So in neutral atoms, protons equal electrons. So helium over here has 2 protons and it's neutral, which also means it has 2 electrons as well. The number of neutrons, however, is not always the same. We can have different numbers of neutrons in different atoms, and those are referred to as isotopes of one another. Ready? Next, we'll talk about specifically electrons. So they determine the bonding properties of atoms. And the opposing charges of the protons in the nucleus and the negative charge of the electrons in the electron cloud are what cause the electrons to orbit around the nucleus, and it's what keep the atoms intact. They are very attracted to each other, which is what keeps them moving around the nucleus and what prevents the atoms from falling apart. It's what keeps everything together. And they are located in different orbital shells around the nucleus, or levels. You can think of it that way as well. The first shell holds up to 2 electrons. The 2 after that each hold 8. After that, it gets a bit complicated, but in this course, we're just focusing on the first 3 orbital shells. So, and atoms are most stable with their outermost shells being full. So that's why they want to, be a part of chemical reactions. That's why they want to bond to other atoms, is to try to create that full outer shell. And some atoms will give away electrons because they have a very low number of electrons in the outer shell. So they'll give those away to atoms that have a higher number of electrons in the outer shell, that thus completing both of their outer shells. Ready? So next we'll talk about chemical bonds. So first we have intramolecular bonds. Later we'll have intermolecular bonds. So sounds similar, but very different. Intramolecular bonds are found inside molecules, and it's what create molecules. The atoms in the molecule are trying to find a way to fill their electron shells so they can be stable. There are 2 types, ionic bonds and covalent bonds, which are shown over here in the picture. Ionic bonds are when electrons are either given away or taken in, right? So over here, we have this atom on the left. It's going to give away the electron, represented by the squiggly line. It's giving away the electron to the atom on the right-hand side. And so the one on the left is giving away, and the one on the right is receiving. So that's an ionic bond. However, in covalent bond, shown on the right, that's the sharing of electrons, and we'll see that in the next slide. So ionic bonds simply give away or receive 1 or more electrons in order to complete their outer shell. Atoms with 1 or 2 electrons in their outer shell, like I was saying earlier, tend to give away their electrons, whereas atoms that are almost full with like 6 or 7 electrons in their outer shell tend to receive those electrons to try to get their outer shell completed. Ions have been created when there is a charge difference between the nucleus and electron cloud. So ionic bonding creates ions. When you give away an electron, you're going to be less negative because you are losing a negative charge. Therefore, you have a more positive charge. If you're receiving an electron, you're becoming more negative. So you are gaining an electron, and thus your ionic charge total is negative. So down here we have NaCl. We'll talk a bit more about this in a bit too, but this sodium atom is going to give away an electron to this chlorine atom. The sodium atom lost an electron, therefore it becomes positive over here on the right. Therefore, it means it has 1 more proton than it does electrons, so it has a positive charge, whereas the chlorine atom becomes a chloride ion, and it gained an electron, therefore the total atom after the ionic bond is negative. So atoms that give away electrons and now have a positive charge are called cations. You can remember that by cats have paws, So it's positive, kind of funny, but that's how I remember it. And then atoms that received extra electrons and now have a negative charge are called anions. And then cations and anions are attracted to one another, forming the ionic bond. Next, we'll talk about covalent bonds, which we talked about a little bit earlier, but atoms can also share their electrons and they count towards both of the outer shells of both atoms. So if the atoms share the electrons unequally, that creates a polar covalent bond, and polar covalent molecules have a slightly positive or negative charge depending on the molecule, and they are considered hydrophilic, which means that they can easily interact with water. If the two atoms share the electrons equally, that's nonpolar covalent bond. And the nonpolar covalent molecules do not interact well with any molecules that have charges. They are also considered hydrophobic, which means that they do not interact well with water either. Ready? Next, we'll talk about intermolecular bonding, which is different than intramolecular bonding. Intermolecular bonding is found between molecules, not within them. An example of this is hydrogen bonds, which we'll talk about in a second. But these two molecules over here, the one on the left and the one on the right, they have intramolecular bonds within each of them. However, the molecules bonding together is an intermolecular bond because it happens between molecules, not within them. So hydrogen bond is an example of intermolecular bonding, holds two polar molecules together, and it usually has a slightly positive hydrogen of one molecule and a slightly negative atom of another molecule bonding together. These are extremely weak bonds unless there are many of them together. So over here on the right-hand side picture, we have hydrogen bond between the slightly positive hydrogen of the leftmost water molecule bonding to the slightly negative atom, oxygen, over here of the bottom water molecule, and it's also happening between the right and the bottom molecules as well. Ready? So next we'll talk about properties of water. H2O is the formula for water. The human body is approximately 50 to 75% water. It is a polar molecule, and it interacts easily with other polar molecules and ions. It is known as the universal solvent, which means it can dissolve the most substances. We'll talk about that shortly. And it is perfect for transporting the body's essential chemicals and excreting waste. It lubricates surfaces of the body, perfect for reducing friction and cushioning the body's surfaces, organs, brain, what have you. And it has a high specific heat, which is what keeps our body temperature relatively stable. So specific heat is basically how the temperature at which a substance heats up. So water has a very high specific heat, meaning that it takes a lot of heat energy to heat up the molecule. So if it had a very low specific heat, our body temperature would change drastically very easily, and that would be deathly. That would be like fatal to us. So it needs to have a high specific heat to maintain our body temperature's stability. Ready? Next, we'll talk about solvents and solutes. So a solvent is the substance that dissolves the solute, whereas the solute is the substance that is dissolved. Making that make a little more sense over here in the picture. The solute and the solvent of a salt solution. So we have the solute is what is dissolved, so the salt. The solvent is what dissolves the salt, so the water is the solvent. And I always think about this like the universal solvent is water, right? Like we talked about before. So I always think of that's what dissolves the other. That's what dissolves the solute. And they combine to form a solution, in this case, a salt solution. So a concentration is the amount of solute that is dissolved in a given amount of solvent. The more the solute you have, the higher the concentration. The less solute you have, the lower the concentration, right? Body concentrations are measured in osmolarity. That's just a vocab word for us to learn here. Ready? Next, we'll talk about pH. So pH is a scale from 0 to 14. The lower numbers meaning the substance is acidic, the higher numbers meaning the substance is basic, right? So it measures the amount of concentration of hydrogen ions in an acid, all right? or in a base. So the stronger the acid, the higher the concentration of hydrogen ions and the lower the pH. The stronger the base, the lower the concentration of hydrogen ions and the higher the pH. So over here in the image, we have 0 representing more acidic substances, right? From 0 to about 6 is a little bit acidic. 7 in the 6 to 8 range is more neutral, but 7 is the most neutral a solution can be. And 14 is the most basic a substance can be, right? And in that end of the spectrum, more bases occur, right? So strong acids and strong bases can be damaging to human tissue, right? So we know that hydrochloric acid, which is represented by a pH of 0 on this chart here, that is very harmful to our skin, right? You might use it in chemistry class, but we always wear gloves, we always wear PPE to make sure that we don't get harmed by that acid. And same with drain cleaner, right? We don't want to touch that to our skin. We don't want to ingest it, obviously, because strong bases and strong acids, not just acids, are harmful to us. So a neutral pH of around 7 and the solution would not be considered acidic or basic. It's considered neutral, right? Our blood has a pH of around 7.35 to 7.45 and is therefore slightly basic, right? So a little bit more about acids. And bases, they dissociate in water into electrolytes. That is something you'll learn a lot about in chemistry if you happen to take chemistry. And they can change the properties of the solutions in which they are dissolved. So acids, they're proton donors, or a substance that releases hydrogen ions into a solution. They have a pH less than 7. An example earlier, we gave hydrochloric acid, and this is the formula for the dissociation of hydrochloric acid in water, right? And then bases, they're proton acceptors, or substances that can bind hydrogen ions in a solution. So pH is greater than 7 are bases. And here we have HCO3 minus, which can bind to hydrogen ions in a solution to make H2CO3. This is the equation for that. Ready? Next, we'll talk about buffers. So buffers are very, very important, especially in the human body. The pH of human blood, as we stated before, is about 7.35, a little bit more. Under healthy conditions, this must be maintained despite constant additions of acids and bases. As you can see over here in the picture, I like this graphic because it shows that a little change in our blood's pH levels can be fatal to the human body. Just going down by 0.35 or going up by 0.35 can cause us to get sick, and even going down by a little more than 1 can kill us. So buffers are what are needed to stabilize pH. They help resist changes in pH and help keep us alive. Blood has several different types of buffers in order to ensure a mostly stable pH for ourselves. And they're very helpful. They are what keep us alive. Ready. Next, we'll talk about salts. Salts are composed of an ionic compound of anions and cations, which you talked about earlier. An example is sodium, Na+, which is a cation combining with chlorine or chloride, negative, right? It's chloride anion to form NaCl, which is common table salt. And that's what is shown over here on the right-hand side. That is an ionic compound that we talked about this earlier, right? When we did ionic bonds, we had the same example here. And we talked about how sodium gave away its electron to chlorine. Therefore, it became a sodium ion and a chloride ion. And the sodium now has a positive charge because it gave away an electron, and the chloride ion has a negative charge because it received an electron. So cations and anions are needed for life. They're used for exciting muscles and neurons, which you'll definitely learn about in Bio 141. They're used for blood clotting and release of neurotransmitters, also learned about more in Bio 141. And most common ions in the body are Na+, K+, so that's sodium, potassium, calcium 2+, and magnesium 2+ as well. And then also chloride. Ready? Next, we'll talk about macromolecules. So there are four types of macromolecules that we'll talk about. First is lipids. Lipids are generally nonpolar. They do not interact easily. There are three categories. First, triglycerides, which are fat used to store energy and insulate the body. They are hydrophobic, which means that they do not interact well with water. The second kind are phospholipids, which are what cell membranes are made of. We'll see a bunch of this later on in the slideshow. And phospholipids are amphipathic, which means they have one polar region and one nonpolar region, one hydrophobic region, one hydrophilic region. That means that they're amphipathic. And the third type is steroids, also known as hormones in some cases. They're made of cholesterol, and the examples are testosterone and estrogen. To talk about is carbohydrates. The monomers, or the smaller building block that build up to carbohydrates, are known as monosaccharides. The polymer of carbohydrates is polysaccharides. So basically, a bunch of monosaccharides form together to make polysaccharides, which form together to make the full carbohydrate. The polysaccharides are broken down to be used as energy, right? When we break bonds, we release energy. Monosaccharides are simple sugars. A great example of this is glucose, right? And they taste sweet, but they can also combine to produce disaccharides, such as sucrose or lactose. And they're mainly used for energy. So they're what the body uses first for energy. So that's why a lot of runners, people that do track, they carb load before a run or a marathon, right? Because that's what our bodies use first as energy. So it kind of elongates their energy stores. Next type of macromolecule that we'll talk about is nucleic acids. Their monomers, or their smaller building blocks, are called nucleotides. They combine to form ribonucleic acid, known as RNA, and deoxyribonucleic acid, known as DNA. Adenosine triphosphate, also known as ATP, is a nucleotide used by cells to do work. DNA is genetic material passed down to you and after you. It is a set of genes or recipes that code for specific proteins, whereas RNA has many forms and it's mainly used for protein synthesis. So over here, you see this double helix on the right, that's DNA, whereas the single helix is RNA. The last type of macromolecule we'll talk about is proteins. So the monomer of proteins, smaller building blocks, is the amino acid. Cells link amino acids in the order indicated by DNA, so it's kind of like a code, and it makes the linear protein. Then the proteins fold to create a secondary structure. They fold some more to create a tertiary structure, and they usually function well after this tertiary structure, but some need the quaternary level of structure, in which they fold upon themselves even more and create that quaternary structure. The structure is very important to function. You'll learn so much about this in Bio 141. Everything in the body has specific structure, and because of that structure, it determines its function. You'll hear a billion times, form determines function. It truly does. If you change the form of something, it changes the function and sometimes makes it even non-functional at all. Next, we'll talk about energy. Energy is the ability to do work, and we have two kinds: potential energy, which is known as stored energy, and kinetic energy, which is the energy of motion. Talk more in depth about that. First, potential energy. Potential energy is stored energy again. So in humans, a most common form of potential energy is the energy stored in chemical bonds. The body also stores energy with concentration gradients across cell membranes, right? So this picture here, the biker's on the top of the hill and he has potential energy because he's not putting in any work to go up the hill, and he doesn't have kinetic energy yet because he's not going down the hill. His energy is stored right now. Whereas kinetic energy is the energy of motion. This can come in the form of radiant energy, also known as heat, sound energy, mechanical energy, electromagnetic energy, also known as light. The image seen here is sound energy, right? You might not believe it, but sound is kinetic energy. It's movement. There are waves in the air in which we hear things, right? The frequency of the waves determines the pitch, and it actually is moving constantly. We kind of take that for granted a little bit, right? We don't really think about that unless you're in a science class learning about it, but it is kinetic energy. Ready? So chemical reactions we'll talk about next. All the chemical reactions in an organism make up its metabolism. Okay, that's a very important word, especially in Bio 141, but you will want to know that the metabolism is made up by all the chemical reactions in an organism. Okay. Chemical reactions begin with the reactants. So the reactants are at least one substance that a reaction begins with. In this equation here, A plus B yields, the arrow means yields, AB. So the reactants would be A and B, whereas the products would be AB, right? The one at the end. The products are one or more substances that a reaction ends with, right? So A plus B yields AB. The products are what's on the right-hand side, AB. Reaction rate is the speed at which a reaction occurs. That makes logical sense. Kind of easy to remember that, but the types of chemical reactions are synthesis reactions, decomposition, single replacement, and double replacement. Those are very common ones, not all of them, but very common ones. We will talk about synthesis reactions here. So synthesis reactions are also known as anabolic reactions. They are chemical reactions that combine substances together. So just like in the last slide, A plus B yields AB, that's a synthesis reaction. It combines substances together. They are also called anabolic reactions, like I stated before. And in this image over here, we have energy and we have a bunch of atoms, and they combine to form that molecule at the bottom. So that's an anabolic or synthesis reaction. Next, we have degradation reactions, also known as catabolism or catabolic reactions. They are the opposite of synthesis reactions, okay? They break down at least one larger molecule into smaller pieces. So if we had the formula, CD yields C plus D, that is a degradation reaction. It breaks down that molecule CD into C and D. Hydrolysis is responsible for breaking down polymers, and you'll learn more about that in Bio 141 definitely as well. But the image over here is a catabolic reaction. We have a big molecule, right? And it's broken down into energy and atoms. We know that when we break bonds, energy is released. So that makes sense. It breaks down the bigger molecule. So that is a degradation reaction. Ready? Now we have endergonic versus exergonic. So when bonds are broken, energy is released. We have endergonic reactions, which are when chemical bonds are created and energy is added to them, right? An example of this is our synthesis reactions we talked about previously. Versus exergonic reactions, energy is released. That's like our degradation reactions. Energy is released when we break bonds. So that makes sense, right? They're opposite of each other, which makes sense why our synthesis reaction and our degradation reactions are also opposites of each other. Next, we'll talk about enzymes. So enzymes control and speed up chemical reactions. They are catalysts. And they are proteins. They end in ASE, that ending ACE. They only work with one chemical reaction, so each enzyme has a particular job. And the metabolic pathway is a complicated chemical reactions broken down into smaller parts that need enzymes to function. The enzymes bind to the reactants or the substrates at the active site. Only bind to one substrate due to its specific shape. So I'll explain that a bit more over here in the picture. We have an enzyme that's in yellow, has a curved surface and a jagged surface, right? The substrate, which is curved on one side and pointed on the other, will fit perfectly into that enzyme's active site. Fits perfectly in there. And it can only bind to that one substrate because of its shape. Enzymes are also sensitive to denaturation, though. They are sensitive to things that can cause them to be ineffective, right? Such as changes in pH or high or low temperatures. And inhibitors can also cause enzymes to not work. Inhibitors block the substrate from binding to the active site. Instead, they bind to the enzyme and sometimes change the shape of the enzyme's active site so that the substrate can no longer bond. Ready? Next, we'll talk about ATP, which is known as adenosine triphosphate. It is the product of cellular respiration, and it is energy. All cells use ATP to disperse energy. It is the only molecule that cells can use to do work, which we know that energy is the ability to do work, right? At the end of cellular respiration, energy is used to attach a phosphate chemical group to ADP, which is adenosine diphosphate, creating ATP, adenosine triphosphate. And that can be seen over here. We have a ribose sugar in the center, and then we have adenine on the side, and then we have 3 phosphates. Adenosine diphosphate only has 2 phosphates, which is why, and to get ATP, we attach another phosphate. Next, we'll talk about cellular respiration. We got a little spoiled earlier, but now we'll talk about cellular respiration. So the phase 1 of cellular respiration is glycolysis. Phase 2 is the citric acid cycle or the Krebs cycle. And phase 3 is oxidative phosphorylation or the electron transport chain. You might hear either one of those names. So step 1, glycolysis, is the first step of cellular respiration. It breaks down glucose in the cytoplasm. No oxygen is needed or used, so it's anaerobic, meaning no oxygen, and 2 ATP are produced. Phase 2 happens in the mitochondria. The citric acid cycle happens in the mitochondria, and a small amount of energy is produced, but so is carbon dioxide. Phase 3, oxidative phosphorylation, or the electron transport chain, happens in the mitochondria as well, just like step 2, and oxygen is used to organize products of phase 2 and produces lots and lots of energy. So about 32 to 38 energy or ATP are produced during cellular respiration. And this process can be seen in the image over here. So in glycolysis, we produce 2 ATP, then we enter the mitochondria, we go through the Krebs cycle, and we produce carbon dioxide, and then we go through the electron transport chain or oxidative phosphorylation, and about 36 ATP are produced. Oxygen goes in, and we also produce some water. So if you see the cellular respiration formula or equation, it has one of its products as water. So it also produces some water in the process. Ready? So next we'll talk about cells. They are the fundamental building blocks of all life. We are made of trillions and trillions of cells. Okay. All cells have a cell membrane and cytoplasm. The cytoplasm is where everything is found, not including the nucleus. It's everything around the nucleus. And that includes organelles and the cytosol. We'll talk about those 2 things later on. And cells have many, many, many functions. They take in nutrients. Most of them can reproduce. They eliminate wastes in the body. They adapt to their environment, maintain an internal environment inside of them, and many other things. Over here, I have a picture that compares cells to Legos. So just like Legos, cells build living things, right? We are made of cells, so are animals and plants, and they are the smaller building blocks of life, just like Legos are the smaller building blocks of these much bigger structures. Ready? So the cell membrane is made up of a bilayer of phospholipids. Phospholipids have 2 ends, a head, which is hydrophilic, face outwards, and tails that are hydrophobic. They face in the middle of the membrane. They are basically afraid of water. They don't interact well with it. And the cell membrane is what separates the outside environment from the rest of the cell. And it's important to talk about selective permeability. So the cell membrane lets some substances in and lets some substances out, but not everything. And that protects the cell. So proteins may be embedded in the membrane, allowing for extracellular function. So in this image over here, we have a bunch of proteins, we have carbohydrates, we have a bunch of stuff embedded in this membrane. One thing I want to point out though is towards the bottom, we have a channel protein. That channel protein is partially what lets things in and out of the cell. So because of these proteins, carbohydrates, these macromolecules that are embedded in the membrane, it allows the cell to have specific functions. Ready. Next, we'll talk about membrane transport. So this is in relation to the last slide. So membranes have to control what goes in and out of the cell, right? Selective permeability. Oxygen, carbon dioxide, and water easily diffuse into the membrane, but larger molecules such as glucose, sodium, potassium, they need a protein's help. They're larger molecules, right? So they don't easily just flow through. So passive transport is when no ATP is needed to transport things across the membrane. It's the diffusion from high concentration to low concentration. Think of it kind of like basically like I think of it like a waterfall, right? It's just from top to bottom. Everything's just falling through, right? You don't need any energy it's not, going from lower to higher concentration, right? It's just kind of flowing through. And osmosis is a type of passive transport, and it's the diffusion of water based on solute concentration. So it always is from a higher concentration to a lower concentration, wherever the water is needed to balance out that cell. Next, we have active transport. It needs ATP because it's going from a lower concentration to a higher concentration. And in active transport, they may use pumps, right? That energy, that ATP is being used to pump things through. And then we have endocytosis versus exocytosis, taking in versus releasing substances. So endocytosis is taking in substances versus exocytosis is releasing them from the cell. Righty. Next, we'll talk about tonicity. So tonicity is solute concentration in relation to the surrounding environment, or outside of the cell. So if the outside environment has the same solute concentration as the inside of the cell, that is called isotonic, right? Iso means same. So think of it like that, isotonic. And if the solution outside of the cell has more solute concentration than inside the cell, that's hypertonic. Water will be drawn out, and that will cause crenation or crumpling of the cell, right? Because when water is drawn out, it's no longer plump, right? For lack of a better word, and it'll shrivel up. The opposite of that is hypotonic solutions. When the solution outside of the cell has less solute than inside of the cell, water is pulled in. And this might cause the cell to lyse or burst. Next, we'll talk about organelles that are found inside cells. The first kind are membrane-bound organelles, which basically just means that those organelles are wrapped in a membrane. So, organelles are found in the cytoplasm between the cell membrane and the nucleus. They have specific functions, which we'll talk about here. Each of these membrane-bound organelles has a specific function. So first, the mitochondria, that's what makes ATP. Lysosomes, they have digestive enzymes that digest damaged organelles. The rough endoplasmic reticulum, you might also see rough ER, and the Golgi apparatus make and package proteins. The smooth endoplasmic reticulum, also known as the smooth ER, make lipids, and peroxisomes use lipid metabolism and chemical detoxification. Ready? And over here we have a picture. The nucleus is the pink and orange circle that's cut open in the middle, and everything around it is the cytoplasm where the organelles are found. Ready? So non-membrane bound organelles, we have ribosomes, which produce proteins, the nucleolus, which produces ribosomes, the cytoskeleton, which is in charge of cell structure and organelle organization, and the centrosomes, which are used during cell division. So you can see a lot of those over here. We have ribosomes, which are found on the rough endoplasmic reticulum, and the nucleolus is the little circle found inside of the nucleus. Ready? So some membrane modifications that help with movement or just general function. First, we have microvilli, the increased surface area for absorption and secretion for the cell. Cilia move substances, example, moving the egg towards the uterus, right? Or moving the sperm to the egg. But also the flagella are responsible for sperm motility, which is moving the sperm towards the egg. And you can see that here. In the third image on the right-hand side of this picture, you see this tail that the sperm has. That is the flagella, and it helps the sperm move towards the egg, right? You might have heard of sperm having low motility, maybe in a previous bio class, but when they have low motility, they don't move to the egg properly. So that's how I kind of think of that, is like thinking of the thinking of the form of something helps you figure out what its function is. They are always, always, always related to each other. So if you need to remember what something's function is, just think of how it's made. Think of its form, right? Good. Next, we'll talk about cell connections or communication between cells. So first we have tight junctions. Substances can't really move between the cells when there are tight junctions present, whereas desmosomes are very strong connections, but not as strong as tight junctions. And then gap junctions are not as strong as either of them. They have pores that allow signals to pass through. So gap junctions are what cells communicate with each other through. And that in this picture over here, the yellow one at the bottom is a gap junction. That is what cells communicate to each other through. Ready? Next, we'll talk about cell division, specifically first mitosis. Mitosis is the most common type of cell division. It has growth, replacement, and repair of cells in its process. And cells divide once in mitosis. It creates two genetically identical cells. The life cycle of cell division includes interphase and mitosis. Interphase is not a step of mitosis. Maybe people in high school or middle school might have taught you that, but interphase is not the first step of mitosis. Interphase happens before mitosis and actually is what cells are in a majority of their life. All right. So in interphase, cells are not dividing, so that's not a part of mitosis. And they have two growth phases, G1 and G2, but the S phase happens between the growth phases, and that's when DNA or the chromosomes are duplicated. And then after interphase happens is mitosis, which is actually the division of the cell. It divides the chromosomes, creates two nuclei, and then we have cytokinesis, which splits the cell itself. So over here on this image to the right, I want to point out that the green portion of the outer circle is interphase. Do you see that, how large that portion is in comparison to the mitotic phase? It's showing that amount, not because there are more phases or anything like that, but that's because the cell spends most of its time in interphase. And then the mitotic phase has prophase, metaphase, also has prometaphase between them, but in this case, it's just prophase, metaphase, anaphase, and telophase. And then it goes into cytokinesis, which is when the cells actually split in half. Ready. So now I'll talk about meiosis, which is another kind of cell division specifically for sex cells. It ends with 4 non-identical cells, which only have half of the parent's genetic material or information. They only produce the male and female reproductive cells, so sperm and egg, or ovum is another word for eggs, right? And over here, I have a graphic that's very helpful, I believe, that it compares mitosis and meiosis. The biggest thing that I recommend you looking at here in this chart is the last boxes. So mitosis creates 2 diploid daughter cells, whereas meiosis creates 4 haploid daughter cells. So that's the main difference between them. They do also have different steps along the way, but the last 2 portions, I believe, are the most important. Ready? We have protein synthesis next up. So proteins are produced through transcription and translation. First, transcription happens in the nucleus. It's when genes are copied into mRNA. They're transcribed into mRNA. You can think of it that way. And then those leave the nucleus. The mRNA leaves the nucleus. And then translation happens in the ribosomes. And the mRNA is translated into a sequence of amino acids, which we know from earlier that amino acids are the monomers of proteins. So, relating everything together, this makes total sense, right? And over here in the image over here, we have the nucleus, which is the darker blue circle or oval, whatever you like, blob, basically. Transcription happens in the nucleus. It transcribes the DNA into mRNA, then it leaves the nucleus and happens in the ribosomes. Now is translation, and the mRNA is translated into amino acids. Ready. Next, we'll talk about chromosomes. So a chromosome is a piece of DNA wrapped around a bunch of proteins. Chromatin is uncondensed DNA, and humans have 23 pairs of chromosomes, all right? One set from the egg and one set from the sperm. And a karyotype shown here in the image is a visual chromosome arrangement. right? So it shows our 23 pairs of chromosomes. The last set is designated as XY rather than, 23, and that dictates genetic sex, right? It doesn't dictate gender, right? We know that gender is different from sex, but it dictates the sex of the offspring, right? So sex-linked traits are carried on these two chromosomes. So we do have 23, the X and the Y. X and Y represents male, XX represents female. Ready? Next, we have genotype versus phenotype. Genotype is an organism's genetic makeup. So we have their alleles, and you probably remember like homozygous dominant, heterozygous, homozygous recessive, all those. And then phenotype is the characteristics that are expressed by the genes, the physical, what we can physically see. So a genotype is an organism's genetic information. So two capital letters, in this case, two capital Bs, represents homozygous dominant. The phenotype that will be expressed because of the homozygous dominant genotype will be purple in this case. Heterozygous, capital B, lowercase b in this case, will also express purple flowers, whereas lowercase b, homozygous recessive will express white flowers, right? The dominant allele is what the dominant portion is always what shows. Okay, now we have Mendelian inheritance. So Gregor Mendel is who discovered that some alleles cover the traits of others, just like we talked about in the last slide, right? The dominant alleles cover the traits of the recessive ones. And we know that homozygous means two of the same letter, or two of the same kind of letter, so two capitals or two lowercase, and heterozygous is one of each, one capital, one lowercase, or one dominant, one recessive, right? Ready? So codominance. So codominance is an alternative inheritance type found in, some flowers, that sort of thing, but also in blood types. So A, B, and O blood types have the alternative inheritance type of codominance, and all the alleles will be expressed. And another example of this, as shown in this image, is flowers. This kind of gives us a visual representation of this. So if we have a red flower and a white flower, they produce a codominant red and white flower. So the red flower has two capital letters, so it is homozygous dominant, and the white flower is homozygous recessive, two lowercase r's. So we get one letter from each parent, and we have a heterozygous red and white flower. Right? So next, we are talking about genetic variation. So during meiosis, crossing over of chromosomes occurs. You can kind of think of it as like they're swapping sections of themselves. Kind of like visually, that's how I think of it. And it results in many variations, but this can cause some issues. Non-disjunction errors can occur where chromosomes do not separate correctly as well. So disorders caused by different number of chromosomes, right? If we have too little or too many, monosomy is when we have one of the chromosomes is deleted in a pair. And that's what's shown in this image to the right. Pair number 9, one of them got deleted. So this person would have monosomy, right? Versus trisomy, which we know as trisomy 21, also known as Down syndrome, is when there's an extra chromosome. And it is sometimes fatal unless it's on a smaller pair. So trisomy 21 happens on the 21st pair of chromosomes, and they are smaller chromosomes. They're not as, they don't have as many codes in them. So it is not necessarily fatal to the human being. People live with Down syndrome. We know that, right? So they, in this case, every single chromosome pair would have 2, but the 21st pair would have 3, have an extra one. Ready. So thank you so much for watching. I hope you learned a lot. I want to, again, bring up the Canvas enrollment link for you to go to that page. It has all this information and more, including practice quizzes for you to practice for your exam. I would definitely recommend it. It's free. You don't have to pay for it, nothing like that. And it is set up by the college. So it's very helpful. So if you have any questions, you may go ahead and drop them below. But thank you so very much for watching, and good luck on your exam.